Okay, welcome everybody. I'm actually not part of the committee, but I'm an intruder because I have the very great pleasure today to introduce Professor Hassan. So, well, it's a great pleasure because I've known him for very long. He's been my PhD mentor, and I have to confess that I think I called him professor, not by first name, twice in my entire life, and both times was when I introduced him to a seminar. So, it's true. So, Professor Hassan is a team leader in Paris at ICM, where he's been for very long also scientific director and deputy general director. And he has a very long, exciting, and productive career in neuroscience. So if we start back in time, he got his PhD in 1996 in the USA at the Ohio State University. And then he stayed in the States for 2 postdocs, I think, and then one, so just long, okay, for 1 postdoc till 2001 when he started his lab at VIB at KU Leuven, and he's been team leader there till 2016, and then in 2016 he moved to Paris as a group leader. So he not only produced awesome science throughout his career, but he also got a lot of scientific important recognition and prizes. I'm just going to name a few. So he received in 2003, he was nominated Young EMBO Investigator. In 2016, he was named the Einstein Visiting Fellow at the University of Berlin. And then in 2016, he received the Allen Distinguished Investigator for his studies that he's conducting in Paris. So his lab is focused on understanding the genetic mechanism underlying brain development and connectivity, and he uses several models to achieve these aims. So he used Drosophila melanogaster, organoid, IPS, and mice. And today he will discuss and share some of his great research about amyloid precursor protein with us. So thank you very much for joining us. It's really a great pleasure to have you here.
Grazie mille, Alessia, for the very kind introduction. It's one of the great pleasures of growing old in this scientific career is seeing your former lab students and postdocs become, start their own labs, and that's really so it's a pleasure to be here. It's a beautiful city, amazing campus. This is my first time, and so I'm extremely delighted. And I'm sorry I'm only spending about 36 hours. So I thought I'd share with you today one of the questions that we work on in my lab that we've been working on for a very long time, pretty much since the lab started, actually, in Leuven, without really meaning to. So the whole story started with a colleague of mine in Leuven at the time, a senior colleague at the time, Bart de Stroper, just sort of came and asked if I'd be interested in looking at, making a fly model for Alzheimer's disease. And I thought, not really, no. And he started explaining about Alzheimer's disease and so on, his ideas about it and so on. And he was talking about this protein where mutations in this gene that codes this protein called the amyloid precursor protein. Is there a pointer or no? Whoops. Cause familial forms of this degenerative disease called Alzheimer's disease. And I started asking, so what does this protein normally do other than being mutated in Alzheimer's disease? And he said, well, nobody knows, but it doesn't really matter because the disease is not related to what the protein normally does and so on. And so I thought, well, actually, no, I don't want to develop a fly model for Alzheimer's disease, but I'd be very curious to uncover the normal function of this gene that you tell me, nobody knows anything about what it's doing, right? And so that's kind of how the story started. And it started with, so before I get to that, a quick introduction about APP and Alzheimer's disease, as you probably all know. The disease, it's a degenerative disease resulting in quite severe dementia in most cases, as in, thank you, Carmen, as many, in many of these disorders, it has a quite late onset. And the majority of cases are sporadic, that is to say, not without a known or definitive causal genetic etiology, although at least in the Western world, there is one gene called ApoE, which is a significant contributor, at least 50%, probably close to 100% of the ApoE4 phenotype are at risk, will develop Alzheimer's disease. But then there's the familial form in which mutations in specific genes are causal, and one of those genes is called the amyloid precursor protein. So the disease was described by this gentleman, a German physician named Alois Alzheimer, in 1906, a famous patient by now called August D. It's the most common cause of dementia well, this is probably an old number. And one of the characteristics of the pathology are things called amyloid plaques, extracellular accumulations of protein, from which the protein then, the amyloid precursor protein, derives its name, and intracellular accumulations of neurofibrillary tangles that are largely composed of a hyperphosphorylated protein called tau. Anyway. And as I said, it's familial and sporadic, and the familial Alzheimer genes are APP and two proteins that contribute to an enzyme called gamma secretase that processes APP, but also hundreds of other proteins. Now, there had, of course, since its identification as the precursor of the peptide that forms these amyloid plaques, there had been a lot of interest in what it does, and people have, mostly through in vitro studies, really, prior to us starting this ages ago now, more than 20 years ago, been implicated in all kinds of things somehow, right? Without really a clear understanding of what the sort of mechanistic aspects behind those different functions in different contexts might be. And I would say that largely this remains, by and large, remains the case, with a few exceptions, as I will hopefully show you today. So the story starts with this gentleman, Martin Leysen, who joined the lab in 2001, specifically to work on this problem, and was later joined by another talented PhD student, Deria Ayaz. Martin was an MD PhD who basically started doing the work, and we started with experiments. I'm summarizing all of their work in one cartoon. We started doing experiments essentially made of overexpressing the protein and then seeing what happens. And basically, we could show that if you overexpress the protein, you get excessive axon growth. And that under specific conditions, and in our case, we used injury to the central nervous system, you would upregulate APP, which would recruit the J and K pathway through its nuclear signal, we reasoned at that time, would also recruit another pathway called the Abelson kinase pathway. Anyway, everything would sort of merge onto the cytoskeleton, and that would drive axon growth. That was kind of the general, somewhat naive model that we had at the time. And I'd like you to keep this idea that APP function regulates J and K signaling in mind for a second. And then so this told us kind of what the protein can do if you put too much of it in a neuron. And the specific thing we were really looking at that time was to drive axon growth in fully formed adult cells. And we could show that, in fact, if you take a neuron whose axon has completed development and you now express APP in the adult, you can drive a bit excessive axon growth. That didn't tell us what the gene did under normal circumstances, and that required the special talents of a special person, whose picture you see here. So Alessia Soldano joined the lab as a PhD student around around the end, shortly after the paper came out, so around 2006, I guess. And Alessia, we decided with Alessia to really go after the endogenous function by trying to figure out what happens in the mutant. Because up to that point, it was described that there are no morphological deficits in the development of the nervous system in these mutants. And Alessia focused on a structure in the fly brain called the mushroom body. And the mushroom body is important for learning and olfactory conditioned learning and memory in the fly, but now we know also visual learning and memory. And it has this very nice super stereotyped axonal structure made of different lobes. You don't really need to remember any of the details. It just basically, there are cell bodies somewhere up here, and they grow axons deep in the brain to this area, where then the branches split to have, if you want to remember anything, it's these things in red, the alpha lobe and the beta lobe. So these would be branches of the same axon growing in two different directions. Okay, so, you know, it was very easy, stereotyped structure, easy to look at if there are any defects. And sure enough, it turned out that in the absence of the fly homologue of APP, called APP-like or apple, so this is a mutant, in a certain % of the time, this beta lobe would simply not grow. Okay, you see here an alpha lobe, a beta lobe, and here you see an animal with no beta lobe. And you know, this was kind of a low penetrance event, but it was pretty clear that this happened and essentially never happened under control conditions. And we thought that this was a really good place to start, because if you have something that's partially penetrant, you can look for other genes whose removal or addition would then make the phenotype better or worse, and in this way identify a pathway that the protein might interact with. And again, long story short, this is taken from a review that Alessia and I wrote after her paper came out in 2013, where basically the main findings of Alessia's paper were where she summarizes what is known about the role of APP in signaling, and where basically what she was able to discover at that time was that the key pathway that seemed to play a role in the growth of the axons in collaboration with APP were proteins involved in a famous, well-known signaling pathway that is deployed across all kinds of cellular contexts called the Wnt pathway, okay? And she identified interactions with several members of the Wnt pathway, notably the downstream cytoplasmic effector called disheveled, but also one of the main receptors called frizzled, and a famous co-receptor called Van Gogh, okay? So, and she was able to identify that these proteins actually physically interact with each other, all right? So that APP was somehow, and she could show that this was also true for mammalian APP, that it was somehow part of a complex that was somehow involved in transducing Wnt signaling, again, down towards this J and K pathway, where presumably all kinds of interesting things could happen depending on cellular context. In our case, we were looking at the growth of axons of post-mitotic neurons, so in that case was, of course, axonal growth. And in concluding, well, no, actually, maybe that was taken from the introduction already. Alessia wrote in this review/perspective, for all these reasons, it may be useful for the field to shift focus to understanding the basic biology and in vivo physiological function of APP, and whether perturbing this function contributes to Alzheimer's disease, okay? So the argument here was that it wasn't just the deposit of this small peptide that came from some part of this protein that is usually in the membrane. It was maybe what this protein did in normal biology that we need to understand in order to get a better understanding of Alzheimer's disease, okay? So given that these proteins are known to play a role in Wnt signaling, the question became, is there some sort of yet to be identified ligand, and does APP interact with a Wnt ligand? Okay. And so this is where another PhD student, Maya Nicholas, who also has now her own professor position, or assistant professor or associate, I don't know if she made it to associate by now, in Cairo, came to the lab and started working with Alessia. And Maya was able to show that, so this is basically what happens when you lose APP. You have 15% of the time you have no beta lobe, okay? Alessia had already shown that if you combine APP loss with Vang loss, you would have about more than 50% of the time this event happening. And what Maya was able to show was that the opposite was true for a protein called Wnt5, one of those extracellular ligands that bind the Wnt receptor complexes, whereby if she removed Wnt5 completely, she would, let me see. Sorry, no, I should start with here. So this is the Van Gogh mutant, which has about 50% loss of the beta lobe. If you now removed Wnt5, you would completely rescue the loss of Wnt5, okay? What this means is that normally Wnt5 would inhibit the growth of the beta lobe, right? But that it was not acting through Van Gogh. So if you removed Wnt5, there's some activity that was now rescuing the loss of Van Gogh. Are you all with me? Right? Okay. Now, you would expect that this receptor of Wnt5 that is still acting in the absence of Van Gogh, right? If you remove it and then you remove Wnt5, nothing would happen because Wnt5, there's no more receptor for Wnt5 to suppress, the loss of Wnt5 to suppress the activity. And what Maya was able to show was that whereas loss of Wnt5 rescues the Van Gogh mutation, it does not rescue the APP mutation, the Apple mutation, suggesting that maybe Wnt5 was the long sought after ligand for APP, and that APP really had a receptor or co-receptor function, because at that time that was still a very controversial notion in the field. And Alessia had actually, before leaving on to Trento to do her postdoc, identified that within the extracellular domain of APP, there were these cysteine amino acids, highly conserved from, between fly and man, highly conserved cysteine residues that are at these regular intervals constituting what is known in the field as the cysteine-rich domain, which is exactly what you see in classical Wnt receptors like Frizzled that you see here. So it looked like, at least at the primary amino acid sequence level, APP on its extracellular domain carried the signature of a Wnt binding domain, okay? And Teng Yuan Liu, who joined the lab much later in Paris, went on to test this hypothesis. And basically, I'm not going to take you through all the biochemistry here, but these are co-immunoprecipitations where the idea is if I capture APP, do I get Wnt5 with it? If I capture Wnt5, do I get APP with it? And the answer is yes, in both directions. If you now chop off this little domain full of cysteines, then the experiment doesn't work anymore, suggesting that there is that APP and Wnt5 interact physically, at least in the context of cells. Teng Yuan was also able to show that in the presence of Wnt5, there was more APP in compartments of the cell that are degradative compartments called lysosomes. So I showed you that Wnt5 seemed to be a negative regulator of the pathway, and indeed, if you add Wnt5, APP goes to the lysosome, and you actually see less full-length APP on the membrane. Teng Yuan was then able to show that this is not restricted to Wnt5. If you take another category of Wnt ligands called Wnt3, which are the Wnt pathways, as you probably know, there's the canonical and the non-canonical pathways. Again, doesn't really matter, okay? They have different types of ligands, but, there's sometimes overlap. It's a very complicated picture. Doesn't matter. The point is that with one type of Wnt, you trigger one type of signaling, and with another type of Wnt, you trigger the other type of signaling. And the interesting thing is that these two types of signaling are in opposition to each other. So if you trigger a Wnt5-based pathway, you downregulate the Wnt3-based pathway and vice versa, okay? And what Tang Yuan was able to show is that in the presence of Wnt3, which also binds to APP, you get more APP accumulation on the membrane, and that is because more APP goes to the Golgi network, where it's preserved from degradation and eventually gets trafficked to the cell membrane. Again, if you don't know the cell biology, don't worry about it. The bottom line is kind of here, which is that APP is some sort of a regulatory member of a complex of proteins on the cell surface that sniffs out the different Wints, and depending on which Wnt is the dominant Wnt at which stage in development, the response is to, so they antagonize each other, and basically the activity of APP slows down axonal growth. This is in now looking at embryonic mammalian neurons coming from the mouse, and accelerates dendritic growth, okay? So in the absence of APP, axonal growth happens faster and dendritic growth slows down in these types of neurons. And the important point here is that this is a balance of which type of Wnt is signaling through the system, okay? Is everything reasonably clear so far? So the only point of everything I've told you can forget all about the details, is that APP, this Alzheimer's disease protein, is a transmembrane receptor involved in signaling during development for neurons to know what to do and the rate at which they need to grow their axons or the dendrites, and therefore, and that we've identified sort of kind of an entire pathway starting from extracellular proteins that are, with an antenna, like a sniffer, like APP, that tells the cell how much of each of these proteins is going on. And it depends on the rate of degradation of APP itself that the cell will react to this extracellular environment, okay? Of course, among many other signals that it's receiving. Okay, so could this be in any shape or form actually relevant to Alzheimer's disease? Now, let's go back to this idea that Alzheimer's disease, the pathology, when you look at the brains of patients, and interestingly, not the other way around. I'll tell you what I mean in a second. If you have a patient that you know they have Alzheimer's disease and they pass away, and then you look at their post-mortem brains, you will find accumulations of the amyloid, of this amyloid peptide. But just the presence of the amyloid peptide does not suffice to prove that a person has Alzheimer's disease, okay? Which already means that it cannot be a causal agent. So the dominant dogma in the field, till today, is that Alzheimer's disease is caused by the accumulation of amyloid peptides. Now, it's a very interesting causal factor whose presence does not allow you to conclude that the disease will happen. But that's a very interesting definition of a causal factor, right?
Is still anyhow a pathological condition, or you can have...
So you can have brains that are filled with amyloid, and the people will be cognitively completely healthy. It has zero predictive power. And of course, you know that nowadays there have been many trials, clinical trials, with anti-amyloid antibodies and various other, all of which have failed miserably. So I think it's pretty clear that amyloid is not a causal factor of Alzheimer's disease. Be that as it may, it is definitely a useful biomarker among others, right? And so we asked, what happens to levels of amyloid if you treat cells that produce amyloid with Wnt? Now remember, what I showed you is the Wnt's affect how much full-length APP is present on the membrane by either degrading it or protecting it from degradation. And sure enough, if you add Wnt3a, which protects APP from degradation, you reduce the amount of amyloid, right? So even though you have more full-length APP, you have less A-beta secretion. And Wnt5 does exactly the opposite thing. And this we see in neurons, in primary neurons. If you try to replicate it in kind of cell lines, like HEC cells or whatever, it doesn't work. So it's a very context-specific, it's very specific to neurons. Okay? So far that we can see at the moment. All right, so clearly that, now, if the physiological function of APP is to measure Wnt signaling and make the cell respond, and the presence of Wnt alters the primary biomarker of Alzheimer's disease, then I think it's not a crazy stretch to suggest that maybe the physiological, as Alessia did in her review, that maybe the physiological function of APP is really what is going on in Alzheimer's disease. So, but this idea applies much more broadly, right? So, in fact, most, if not all, genetic forms of neurodegeneration arise from mutations in genes that are involved in basic biological functions, as I just showed you for APP. Presenilin is a gamma secretase, is a membrane secretase that cleaves hundreds. Probably every single pass transmembrane protein in the cell is cleaved by gamma secretase. And yet we're supposed to focus only on APP. You know, why, right? But think about Parkinson's disease. You know, many mutations in genes that are involved in mitochondrial biology, in DNA repair, in this, that, the other, very basic so-called housekeeping genes, right, are what we find mutated. Now, almost all these genes in the human, at least, are ubiquitously expressed in time and space. And people, of course, in these conditions, genetic conditions of neurodegeneration, are born with these mutations, right? It's not that they acquire them when they're 80 years old. But we're still supposed to believe that neurodegenerative diseases, right, that these mutations have no effect throughout your entire life, right? A Wnt receptor, a mitochondrial control protein, a DNA repair protein, does nothing to you throughout your entire life, at least to your nervous system, until an age beyond which we never evolved to live to begin with. You know, when our species evolved, neurodegenerative disease did not exist because nobody lived that long. So there's no selection on these mutations or on these, you know. There's something that just doesn't make sense. And the alternative idea is that neurodegenerative diseases, as clinically defined by the appearance of clinically measurable phenotypes at late stages of your life, are actually just late onset neurodevelopmental diseases. That is to say that the pathophysiology is present from the beginning. It's just that the system is sufficiently robust to not show those as outward clinically measurable phenotypes.
Look, I'm a physicist, so I'm very prepared for...
No, please.
So, but you know, the idea that these amyloid plaques, they develop, I mean, they accumulate over time.
Yes.
So the idea would be simply, I mean, it is produced along your life, but they reach a critical mass.
Completely. Yeah. So, except, again, they can accumulate in healthy patients, and so that rules them out as a causal factor. That's number one. Number two, many other neurodegenerative diseases show accumulations of other types of proteins, and many genes that have been identified. Again, Parkinson's disease is a great example because by now we have like more than 100 genes that cause familial forms of Parkinson's disease. Almost all of them are involved in really basic cell biological processes. People are born with these mutations, and you know. And of course, I'm not mentioning it here, but there are juvenile forms of neurodegenerative diseases. I mean, there are a few extremely rare cases of Parkinsonism at the age of 6 and 9. Obviously, this was a neurodevelopmental problem, right? So the second interesting thing is this is a kind of a curated list where gene associations just from taking the literature, right, and then making predictions about whether a specific gene might be associated with neurodevelopmental disorders. This is a kind of a resource that was created by Thomas Bourgeron at the Pasteur Institute, who's leading figure in neurodevelopmental genetics. And you find APP as a candidate neurodevelopmental disorder gene. Okay, just based on looking at the literature, right? So the literature itself is suggesting maybe something's going on. And finally, although to be honest with you, this we stumbled upon after we had started the studies I'm going to show you next, but anyway, for the sake of the talk, it works well to introduce it here. Finally, there is at least one case in the medical literature of a child, 20-month-old child, with microcephaly and other associated defects and neurodevelopmental problems, who inherited from the parents a stop codon in APP, and where barely 10% of the protein was left. And in this child, this is the only homozygous mutation, so likely to be the cause of both the intellectual disability and the microcephaly. And so all of this suggests that APP really is playing an important role in neural development. And so we wanted to look at this problem, and we started by querying existing single-cell RNA-seq data of the human fetal cortex, whether APP is expressed during the development of the human fetal cortex, and if so, in what cell types, and what was the pattern of that expression. And what we found is that APP is initially quite highly expressed in neuroepithelial cells, and it undergoes a sharp decline when those neuroepithelial cells become radial glial cells. So as you all probably know, the development of the mammalian cortex, including the human cortex, starts with a cell type called the neuroepithelial cells, which align the neural tube, the ventricle, right? And shortly thereafter, these cells become specialized and grow processes, and that's why they're called radial glial cells for historical reasons, really. But these are the neural stem cells, and so these are the cells that are going to produce all the cells of your cortex, neurons and glial cells and so on, okay? And so when they switch from neuroepithelial to radial glial morphology and phenotype, that is when you know they're going to start to produce neurons, okay? And then, so it dips and then goes back up in intermediate precursor cells. So these are the cells that will divide one last time to make neurons. So not only is it expressed, it has this sort of temporal dynamic, right, where it suggests that it needs to go down when the cells want to start producing neurons. And we verified this on these sections of the human fetal cortex. I believe this is gestation week 14, where you see this is APP RNA, and you see APP strongly expressed in these cells down here that are called apical radial glial cells. So these neural stem cells that line the ventricle of the developing brain. So what we did was to take iPS cells. You all know what iPS cells are, right? So these sort of naive, reprogrammed stem cells from humans, in this case, normal humans. And we used a technology called CRISPR-Cas9 to delete APP, okay? So to make human stem cells that have no APP and ask what happens if you now try to mimic human cortical neurogenesis in the absence of APP. Okay, is this clear? Please don't hesitate to stop and ask any question, even if you're not a physicist. Okay? No, just APP, not APLP1, APLP2. And as often happens in preceding those moments of that we all live for as scientists, those rare moments of discovery where you go, this cannot be true, right? The student who was working on this, I'll show you a picture later, Dania Shabani, you can see this is essentially a story of women in my lab. Right? She put these iPS cells, these stem cells, turned them into cortical progenitors, and, then added something in the medium to make them to start making neurons, right? And you go in and you basically, human neurogenesis is extremely slow, and that's really important to know, okay? And so after a week in this medium that promotes making neurons, normally you don't have neurons, okay? It's too soon. And she came back and she said, there's something wrong with the APP mutant cells. And took these pictures in just like DIC and said, look, you know, these are the control cells. They're very nice. They look like progenitors, and they, you know, they're sitting there. And look what's happening in the APP mutant. They have this weird morphology. I think they're becoming neurons. And I said, no, this is impossible. I mean, you did something wrong. This cannot be, blah, blah. So I said, look, if you're right, it's very simple. We stain for neuronal markers, and if we see them, then that maybe is right. And we did that, and we, of course, generated many different clones. I'm showing you data from one clone, but many different clones and 2 different genetic backgrounds, et cetera, all the controls. And this is a marker of immature neurons and intermediate precursors, extremely rare in the control. You have like crazy in the knockout. This is a marker of maturing neurons, where you see this is TUJ1 or beta tubulin, and you can see it in a few cells that have become neurons in the control, and you see the knockout is full of them. And this and a series of experiments, I was like, okay, I'm convinced now that this must be true. So basically, in the absence of APP, progenitors that are supposed to very slowly make neurons are making neurons like crazy. And then, just to make sure I would believe, because this was extremely surprising. Nothing in the literature suggested like something like this would happen. And I still didn't want to believe it. So I had a different student do a different experiment, which is the following experiment, which is to take these mutant and control progenitors, sparsely infect them with a GFP-containing virus, and trace what happens to GFP-positive cells over time. And Marwan Benaisa, a Spanish master's student in the class at that time, in the lab at that time, basically, so the experiment is, you know, this progenitor will divide, it will make a lot of cells, they will all be green, and then you can ask, among these green cells, let's say after 1 week versus on the first day, how many are progenitors and how many are neurons, right? And you can calculate the ratio, and you can divide it by the number of starting cells, and you can know what is the potential of one progenitor to generate in the future. Now, as you would like for a normal, well-behaved, polite human progenitor to do, after 1 week, one progenitor generates 8 other progenitors. Okay, so mostly what it's doing is proliferative divisions, right? It likes itself, so it makes more copies of itself. And every now and then it spits out a neuron, right? Because why not? If you now look in the mutants, one progenitor generates only one progenitor after a week, but generates somewhere between 15 and 30 neurons. Okay? So normally, within the first week, progenitors make more progenitors, and every now and then a neuron escapes. In the absence of APP, the progenitor loses proliferative divisions and undergoes what you would call self-renewing divisions. So it makes one copy of itself and one copy of another cell that then goes on to make 2 neurons. And then again, one copy keeps that one copy, but keeps making neurons. Okay? Now, we started to ask, what exactly is going on here? So they're making neurons at a much faster rate, right? Are they making random neurons? What's going on to the neurons? And the answer is nothing. After 15 days, you see the appearance of a marker of early born neurons. Okay? So you know that the neurons in the, I'm sorry, I realize I missed a very important introductory slide, which is how the cortex actually develops, right? So basically, you have these progenitors, and they make different sets of neurons that are born at different times, and those different neurons sit in different layers, okay, and express different markers. And we call them early born versus late born, or deep layer versus upper layer cells. Okay? I apologize for not putting that in. So CTIP2 is one of those markers of early born cells, and it appears about 15 days in culture, and the same is true in the knockout. It's just a lot more of them. If you look at a late born marker, it's not there yet. At 30 days, you start to see the late born marker of neurons in both. You just see it a lot more because there are a lot more cells in the knockout. So the order with which the neurons are produced is the same. There's just a lot more of them. Now, at 60 days, you ask, do I have markers, are these cells differentiating sooner, right, these neurons? And the answer is no. At 60 days, they don't express any synaptic markers. By 120 days, now they express synaptic markers in both the control and the mutant. And by 160 days, you start to see activity in these, in this case, in these organoids, okay? So you have a sort of an acceleration of neuronal production without any change to the order of the production, the temporal order, okay? Not the temporal rate, the temporal order, we'll get back to that in a second, nor to the rate at which the neurons then differentiate, okay? So the mothers are in a hurry to make neurons, but the neurons then take their time to develop them, okay? And that tells us something very important, which is the clock of the progenitors and the clock of the neuron are two different clocks, okay? So even a neuron born too soon will then go back to differentiate what we would call at a human time scale. Okay?
What about the structure? I mean, the structure of the tissue that you find at the end, is it similar or is it?
Well, I mean, this is all in vitro, so there's really no structure to speak of, and that's a very good point. The prediction is, of course, not, because if you produce neurons at a much faster, but maybe I'll get back to that in a second. Yes?
Sorry, may I ask a question?
Yeah.
You said the temporal order along which neurons are generated, different neuron types are generated is normal.
Yes,
My question
as far as we could see.
my question is the onset of the production of each kind of neuron is untouched, or is it anticipated a while?
Yeah, this is a great question. We haven't really yet looked at this level of detail in the mutant. So whether the time, we tried to do that with BRDU, EDU tracing. The problem is that labeling with BRDU and then waiting for the markers to appear, it's super toxic. So actually most of the cells died before we were able to catch when are they born exactly in terms of. But I'll come back to a kind of a similar experiment in a second. So progenitor clock and neuron clock are genetically separable and likely therefore independent, and that APP seems to be specifically required in the human for the progenitor clock. But essentially, these data can be interpreted in two different ways. The first is that we have basically what we would call interrupted neurogenesis. That is to say that the progenitors are producing all the neurons at the right time, but because they're switching from early on from proliferation to neurogenesis, at some point you run out of progenitors, okay? So you produce a kind of a subset of deep and then upper layer cells, but you don't really produce enough upper layer cells, okay? Because you run out of fuel, essentially, and the whole process stops. Now, this makes a very specific prediction, that at the end of the experiment, if you measure the early born to late born ratio, it should be very high in the mutant compared to the control, right? Because you don't make late born cells. And then eventually, if you wait long enough and you do another experiment here, right, they should reverse, because by now, right, in the knockout, there will be a low early born to late born ratio because the controls kept going, right? The other model is that you have accelerated neurogenesis. That is to say that the knockouts, what they do is they run the whole program, they just run it on a shorter time scale, okay? And to give you a metaphor for that, the difference would be you're driving from here to the square in front of the Gulf, except one is driving a Panda and the other is driving an Alfa Romeo. Now, obviously, one's going to get there sooner than the other one, right? But they both get there, right? Just one gets there slower, and the other gets there faster. So it's not about the fuel, it's about running the program on an accelerated timeline. That is to say, it's about the genetic control of time, okay? The fact that what the progenitor is doing in this model, right, is encoding, it knows how much time it has, it just decides that that time is shorter than normal. Now, this model makes the exact opposite prediction, that if you stop the experiment here, there will be a low early born to late born ratio in the knockout compared to the control, but eventually the control will catch up and the ratios will be the same, right? So in fact, measuring this ratio, because the two models have completely opposite predictions, measuring this ratio will tell you which one's going on, and then measuring it at the end will tell you whether they reverse or they become unequal. And the data were pretty crystal clear. So if you look at that ratio early on, it's higher in the control, so lower in the knockout than in the control. And if you keep tracing, in the end, there will be no significant difference between them. Okay? Which means that this is the correct model. Which means what's going on is that knockout human progenitors that lack APP produce their entire neurogenic program on a faster time scale, essentially by making neurons instead of making copies of themselves. So of course you end up with a smaller pool, right? And therefore, because you have more time here, that might explain why you get microcephaly in this child. So it generates all the right ratios of cells, but not the right numbers of cells. See? And this is really interesting from the perspective of starting to kind of dissect how development is encoded in different ways, how different aspects of development can be dissected, teased away from each other, right? The fact that you have a genetic program that you can run at different time scales, right? The fact that ratios and numbers can be controlled separately from each other. And it's all, I think, worth thinking about.
Sorry, ask another question.
Yes, please.
I'm sorry. But did you observe any change in the average length of the cycle? Or
Great question. Yes, we did. Actually, one of the reviewers asked about this and said, well, maybe this is trivial. Maybe the reason this is faster is because the cell cycle is a lot faster. And if anything, in the mutant, the cell cycle is slightly longer. So it's not about the cell cycle. It's really about the switch of the mode of division of these cells. Now, we did a whole bunch of experiments that I'm not going to go through in detail. We did single-cell RNA-seq, bulk RNA-seq, and of course our own knowledge of our own literature, so to speak, to suggest that maybe this, to ask basically the question, is this phenotype, right, based on the molecular mechanisms that we've already discovered in completely different contexts, in the fly and then in the mouse, right? And the answer is yes. So it turns out that what's going on is that APP is acting as a Wnt3 receptor, and Wnt3 is known to be a promoter of proliferative divisions of stem cells of all kinds, including neural stem cells. And basically, in the absence of APP, there is less Wnt signaling, and Wnt signaling is there to promote the progenitor state. But there is also excessive amounts of J and K signaling, where it seems in this context, APP, and this had been shown in previous papers, APP can slow down or inhibit J and K signaling, which then activates a transcription factor called AP1 made of two subunits called Jun and Phos. Okay, and we could show that we had too much Jun, too much Phos in the absence of APP, and they were phosphorylated, et cetera, et cetera. And that Jun and Phos were driving neurogenesis. And so simultaneously, you had a reduction in the capability of the cell to stay in a proliferative progenitor state, and an increase in its capacity to undergo neurogenesis, right? Now, what this model suggested is that these were sort of, the presence of APP gates the interaction between these two pathways. Because you can imagine under normal conditions, what you want, right, is to ensure that if you're in a progenitor state, you're not undergoing neurogenesis. And that when you're undergoing neurogenesis, you're reducing your proliferative state, right? So having a single protein that bifurcates to regulate this balance, to read the environment and regulate the balance, is kind of a useful thing to have, right? This is how you couple. What would be otherwise completely different processes are coupled in their response to the external environment by the fact that you have one sniffer, so to speak. And this model makes a very important prediction, which is that if we were to interfere downstream of APP, we should be able to separate the effect on the progenitor state from the effect on neurogenesis, right? And so this is the experiment. This is an experiment where we use a pharmacological inhibitor of AP1, which prevents the dimerization of Phos and Jun. And what happens in this case is we read out the neurogenic state of these progenitors through a protein called neurogenin 2. Essentially, apical, human, not just human, mammalian neural progenitors, when they want to differentiate, they express a transcription factor called neurogenin 2. Okay? And you can see in the absence of, in wild type, very, very few cells at this progenitor stage express neurogenin 2. Maybe this one up here. In the knockout, a lot of them express high levels of neurogenin 2. If you inhibit AP1, you go back to a wild type state, you see. And then, indeed, the cells don't differentiate, so they don't express this early born neuron marker double cortin. Okay? You can see this here. Now, this is for double cortin, this is for neurogenin 2. On the other hand, if you count the number of cells that express the progenitor marker, they are not rescued. Okay? So you're still not undergoing proliferation, but you're stopping neurogenesis. If you do the opposite experiment and you pump the medium with Wnt3a, trying to bypass the requirement for APP, because the other Wnt receptors, they're still there, right? Then you have the exact opposite effect, which is that you rescue the, you rescue the proliferation of progenitors. Okay, so proliferation goes down, progenitors go down. Now you can bring it up with Wnt3a, not quite to wild type levels, but a pretty significant rescue. But you see no effect on the fact that these progenitors go on to make neurons anyway. So you make them divide more, but they make neurons, right? So showing that, in fact, you can separate the two pathways downstream of APP by interfering with them separately, and showing that, once again, progenitors use APP to estimate the environment and therefore react accordingly, but that downstream of that, other regulators can regulate the different processes quite separately from each other. And so this is, again, the model. This is Dania, who did all the work, and this was published recently. And so suggesting that through the regulation of the balance between a progenitor state and a neurogenic state, right, APP is one of the proteins that contribute to the control of the prolongation of this, the famous ontogeny of human cortical development, right? The idea is that something happened during evolution to take something that can happen within a week in a mouse and make it happen in about 100 days in a human, and yet fundamentally the same thing happens. That is to say that you produce different types of neurons in a specific order at different times, and the mouse neocortex and the human neocortex, I mean, despite a lot, other than their size and the amount of cells they contain, don't really have massive fundamental differences. It's not like we have a completely different cortex, right? We have fundamentally the same cortex, which for some reason develops a lot slower, giving in part, this is the reason why we have a ton more, 1,000-fold more neurons in our cortex than that in a mouse, right? And so something must have happened during evolution to expand this time scale to allow, in particular, at later time points for the large amount of generation of what are called upper layer neurons in primates and in humans in particular. And the argument here is that the co-option of a very ancient, so a lot of the work trying to understand this evolutionary transition assumes, and some of it correctly, that this must have been due to genetic innovations in the primate and human lineage. That is to say, new genes or new copies of genes or whatever. What I'm showing you here is that a very, very, very ancient protein has been co-opted. It doesn't seem to play this role in either mouse or fly, but has been co-opted, probably due to other factors that are also slowing down human neurogenesis, to now participate in this particular mechanism using nothing new, using the same thing it does in other species, binding to Wnt and regulating J and K. Okay. So let's go back to Alzheimer's disease for a second. Now, what I showed you is the effect of removing APP on human cortical neurogenesis, right? That is not what the patients have. That's what this one child in the literature, probably others too, not described in the medical literature, had, and that's important, but that's a neurodevelopmental disorder. And I've been arguing to you that maybe neurodegenerative diseases, maybe particularly Alzheimer, has something to do with development simply because APP has something to do with development. Not exactly the same thing. So what we've been doing recently is asking, what if we made human cortical progenitors and studied human cortical neurogenesis from patients with Alzheimer's disease, from iPS cells from patients with Alzheimer's disease, of course with the appropriate genetic controls. So this is very preliminary. This is the work of Johnny, a very talented postdoc in the lab. And Johnny asked, do mutations that cause Alzheimer's disease in APP have an effect on the rate of neurogenesis, of human cortical neurogenesis? And is that effect similar to the knockout or something else? And the reason we asked about the something else is it is known that there are extremely rare cases of familial Alzheimer's disease that are due to an extra copy of APP, so a duplication of APP, and others that are due to mutations in the promoter that give rise to about 1.2 to 1.5-fold more mRNA expression. And the argument had always been, well, of course, you have more APP, so you make more amyloid. And I showed you that that doesn't necessarily work that way. And so in taking patients from, we actually were looking at two different mutations. One is a duplication of APP, and the other is the very famous Swedish mutation. It's a point mutation that is a very severe case of Alzheimer's disease. And we find the same thing. Under control conditions, most of the cells, before we start neurogenesis, of course, express SOX2, so they are progenitors, and rarely express neurogenin 2, as I showed you, but still, you have about 0.434% of the cells that are neurogenic at this stage. Even that tiny amount in the AD mutation is suppressed. Okay? Now, you look 30 days later, where, of course, a lot of cells in the control express neurogenin 2, and ergo fewer express SOX2, and you can really see it on the image. See, by 30 days, you've lost about 50% of the progenitors, because now they're making neurons, whereas you have at least 10% more that express SOX2 in the AD mutant, and about twofold less that express neurogenin 2. Okay? So at a time when you're supposed to be neurogenic and producing neurons, the APP, the Alzheimer's disease IPSs are just waking up from their proliferative slumber and go, oh, maybe now I start to make some neurons. And you can follow this over time, and you see that at some point, about 2 weeks, within a week actually, you stop losing progenitors, or you slow down the decrease in progenitors, which is very beautiful and steady in the control, in the AD case, always in red here. And you don't really accumulate, I don't know what happened at day 15, but you don't really accumulate more neurogenin 2, whereas of course you're accumulating very, very quickly, as I showed you just within a month. And if you now look at the production of neurons, right, within 2 to 4 weeks, 10% of the cells in these cultures are neurons, okay, whereas less than 5% are neurons in the AD. So you have exactly the opposite effect to the loss of function, which is a slowing down of the production of neurons. And we're now in the process of making 3-dimensional organoids to look at this and looking at different cell types and their rate of production and so on and so forth. I can't tell you more than this at this stage, not because I don't want to, but because we don't have. And so the question is, might there be a slowing down of cortical neurogenesis in Alzheimer's disease? And if that is the case, right, what is that? Because everything I'm showing you here is excitatory cortical neurogenesis. That is to say, the production of glutamatergic excitatory neurons in the cortex is what we're modeling. But if you change the numbers of excitatory neurons, that's by definition meaning you're changing the neuron to glial ratio. You're also changing the excitatory to inhibitory ratio. Which means, of course, this is all speculation on my part, but that means that obviously, you know, by the time you're born, you probably have enough of every cell type to make it, except that throughout your life, you essentially have a vulnerable cortex that has never been working quite right. And so the idea is, over time, it's not the accumulation of the amyloid peptide, it's the accumulation of dysfunction of the cortex that eventually just gives up. The metaphor for this would be if we go back to the panda and the Alfa Romeo, and we say, well, you know, let's say we make them drive at the same speed, right? But we fill the Alfa Romeo with 70% gas. Now, for 70% of the way, you're not going to see any difference, right? At 70% of the way, the Alfa Romeo is going to break down and the panda will keep going. Now, nobody in their right mind will tell you the problem arrived 70% of the way. The problem was there when you decided how much gas to fill, right? And this is the kind of metaphor I have for neurodegenerative diseases. That people who will suffer from neurodegenerative disease in the future, at least under genetic mutation conditions, start life with a 70% tank full of gas in their head, and of course they will run out of it earlier. Okay? It cannot be the only factor, and I'm not saying that, I'm saying it's just a different way of thinking about where the problem starts, because if we're right about this, and I'm not the only one in the field to start to think about these ideas, that would mean that we might be able to identify biomarkers of dysmorphism, if you will, of the generation of cortical cells and perhaps other cells in the nervous system to start to say, well, this person is at risk. And maybe we need to start warning them or giving certain treatments or lifestyle changes much earlier. Because honestly, you don't need to treat neurodegenerative disease. You just need to delay it by about 20 years. Because then we'll all be dead before we have it. So, you know, I mean, death is an option as a treatment, right? So, big picture, blah, blah, which Danya and I recently argued about in this perspective, and we published in Development, is this, as I told you, most of all, if not all, genetic forms of neurodegeneration arise from mutations in ubiquitously expressed genes with basic biological function. But we also know that different functional subtypes of neurons develop at different times, and the peak of neurogenesis is different in different regions in the brain. And so perhaps different neurodegenerative genes have different temporal profiles of expression or activity, which might explain why mutations in one gene give you a cortical disorder and a mutation in another gene gives you a midbrain disorder, and so on and so forth, right? Even though they're all expressed everywhere, right, when we look in the adult. But during development, no one's taken the time to do a careful analysis of the spatio-temporal expression of so-called ubiquitous housekeeping genes, right? And I showed you, with APP, if you were to look kind of globally, you'd say, ah, well, expressed in all cells all the time. But you just look at different subtypes, and you see that during a very tiny window of development, it goes in a single cell cycle, right? If the cell's going to be neurogenic, it drops. If the cell's going to be neuroepithelial, it stays high. And so perhaps when matters. And it's unfortunate the physicist was asking questions, because in fact doing those experiments in my lab got me completely obsessed with the notion of time. Actually, it started by looking, okay, what is time in biology other than circadian rhythm, right? And then you find very, very little, you know, it's an emerging field. And then you go from what is time in biology to what is time. And little did I know what a battle this is in theoretical physics. Is time real? Is it emergent? If it's real, is it emergent? Is it real at all? Einstein would tell you no, time does not exist. The equations of quantum physics are completely time symmetrical. And in theory, therefore, they can go backwards and forwards in time the same way. But our experience of time clearly has an arrow. And, you know, there's beautiful arguments, you know, from Carlo Rovelli and Ilya Prigogine, who wrote, you know, The End of Certainty. Anyway, so I started reading all these books on theoretical physics just from doing biology experiments on APP. And so, you know, I think this is a great institute to come visit as often as possible. Hint, hint. Okay. And so temporal differences in brain development obviously have an evolutionary origin, and this may help explain both differential phenotypes of different neurodegenerative diseases and the particular vulnerability of the human brain to them. It's practically impossible to make a mouse model of Alzheimer's disease. You can make a mouse model of overexpressing amyloid. That's not Alzheimer's disease. And if you make exactly the same mutation, the mouse says, thank you very much, and goes on with its life. Okay? So I will stop here. I probably went way over time. I apologize for that. I mentioned all the people who were involved in this. I want to thank especially Alessia for actually starting us down the road of WINT and APP. And of course, all the funding, past and present, and you know, my institute, which is a fantastic place to work. And thank you all for your attention. Yeah, yeah, yeah. Mm-hmm. Yeah. You mean in the APP condition specifically? Yeah, yeah. Yeah, yeah, sure. No, of course that's entirely possible, and there are papers out there suggesting that also in the Presenilla mutations there are neurogenesis issues in, you know, similar models. And it's entirely possible based on what I showed you in the mouse, at least in vitro, in these neurons, when we were looking at the growth and branching of the axons. I didn't show you all the details, but basically, if you look later, right, the knockout and the control, they look the same. But the rate at which they arrive there is very different. So in the APP knockout, the axon is growing and branching faster, actually, in the mouse. We didn't look at this in the human very carefully because we found this spectacular phenotype early. But defects in circuit formation under the disease conditions can happen from different perspectives. The first is if you do have a change in the temporality of neuronal production, then of course, you know, many of these cortical cells in specific layers will project either to the other, to, you know, cortical projections or corticothalamic projections. And so if they are delayed for example, in the patients, in their formation, then they might arrive at different times in the areas, in the target areas, and therefore have a slightly different innervation pattern. The other thing that I alluded to is the change in the excitatory inhibitory ratio, because if inhibitory cells that are produced in the ventral telencephalon are invading the cortex at a time that is where the cortex is still undergoing neurogenesis or doing it slower or different types of, then the way these cells and people like Gord Fischel and Oscar Marin and many others have shown that the differentiation of inhibitory interneurons really depends on the local environment in which they invade. And so if that environment is changed because of the rate at which you're producing the local neurons, then presumably those cells will also maybe many of them will die before they integrate and so on. So you can change the circuitry in that way as well. And then finally, you can have indeed the cell autonomous effect of how fast is the axon growing versus, and the interesting thing is that the effects on axon dendrite are exactly opposite. So the mutation which accelerates axonal maturation is slowing down dendritic maturation. So it's another way you can affect the formation of the circuitry. So the idea would be that there is a plethora of subtle changes in these patients, right? None of which are big enough to make a brain not functional, but all of which together make the brain more vulnerable over time to overuse, basically, right?
And the other curiosity is regarding the fact that the mouse doesn't have these defects. Do you think it's due since the molecules are all there?
Yeah, absolutely.
Do you think that it's because their clock is short?
Yes. So
There
are two possibilities, right? The first is that exactly that. That is to say, obviously APP is there and Wnt is there and everything is there, right? The first is that indeed, because due to other factors, partly their metabolic rate, as Pierre van der Hagen recently showed, they are less vulnerable to that is to say that the human system becomes vulnerable to this imbalance between APP and Wnt signaling because of the slowing, and when it becomes vulnerable, it feeds back, and then you get this acceleration, right? The other possibility is that it's just not expressed high enough for in the appropriate part of the cell or whatever to have, and there really is no effect on the mouse. The third possibility is that there is an effect in the mouse. It's just because we're looking at an already extremely rapid rate of neurogenesis, we didn't really see it, right? Because the way we looked at this was to do neuronal stainings too soon, and then all the marker stainings when you're supposed to get them, and everything looked normal quantitatively. That doesn't mean that if you don't have a 10% difference that you're actually not going to see it. So we're gearing up to do single progenitor clones in vivo now, using the MADAM approach in the knockout background. Because, and as Carmen pointed out to me later, maybe what you would have in the mouse is what happens later is the production of glia. So maybe, yeah, neurogenesis doesn't get significantly accelerated, but then if you have an acceleration of gliogenesis, then you start to see the effect. So by looking at how many different cell types and how many of each cell type a single progenitor produces during mouse cortical neurogenesis in an APP knockout, we should be able to address that question as to whether there's really zero effect in the mouse, or whether the effect is not on neurogenesis, but on growth and axons and so on, or glia or whatever. So.
Well, I have a couple of questions. One is related just to this glia issue that you mentioned. So again, do you have any hint that the activation of gliogenesis is anticipated?
Not yet. Not yet. We haven't really looked at the production of astrocytes and oligodendrocytes in those knockouts. That's something we're going to do with the AD mutants, because now they seem to be slowing things down. So does this mean that we, now are going to delay when glia appear, which would, of course, also be a potential contributor to the homeostasis and the health of the cortex that you produce? We don't know the answer to that.
And the second question was a little bit more specific. So it's about how the progression of time is encoded in these neural stem cells. So someone suggests that some stepwise progression of some molecules, including the microRNA, micro-9, for example, could be somehow involved in taking trace of the progression of time.
Yeah.
Now, my question is, do you have any data about the slope at which Mir9 progresses, so relative expression of Mir9 progresses?
We haven't looked into that. So one of the things we're going to do, I mean, I can go back and look at the single cell data that we have and see. We did the single cell only in progenitors because that's where the problem is, but we can look at whether in different clusters of progenitors Mir9 is differently expressed. I never thought about it. No, we didn't look at that. So I don't know anything about Mir9.
It would be really interesting.
Yeah, that would be super interesting.
Because usually it's suggested that for every cell cycle you have a little bit of increase.
Yeah. Personally, I'm not convinced of this idea of a single molecule timer. But God knows I've been wrong many times in my life. So could be wrong. I could be wrong about this. The question you asked that is behind the question, which is the genetic encoding of time, is really, I think just like in theoretical physics, that is the biggest question in biology. How clearly the genome, evolution wouldn't be possible. If the genome was not responding to, even if time is just an artifact of thermodynamic, the second law of thermodynamics or whatever, right? And it's still, evolution is measuring it and playing with it in order to produce those. So if you want to say that time in biology is an emergent property of cellular behavior, then you don't need a timer. All you need is what I showed you, the change in behavior, right? So this very simple equation where speed, right, is basically space over time, right? And if you think about space as the total amount of stuff in a cell, right, and speed as the rate of different reactions, and if you assume that in evolution the rates of biochemical reactions haven't changed, in fact, there's no reason to think that a given enzyme coming from a mouse or a human has a different rate of reaction, right? Then essentially what determines time is space. That is to say, more stuff you have, slower things will go, all right? And so from that perspective, the total amount of material in a human cell compared to a mouse cell, compared to a fly cell, the size of the genome, the redundancy in the genome. It's like an algorithm trying to solve a problem with more information, right? It'll take more time to solve it. So that would be one option. The other option would be that the genome itself is a temporal encoder. In other words, if I'm going to make RNA out of DNA and the more I fill my genome with material, right? In a sense, that's how I'm encoding time. Okay? And then that blurs the notion between the difference of time and space, because it doesn't matter anymore, right? The origin could be temporal and you fill it with space, or spatial and you fill it with time, and then It doesn't matter anymore from that, from that biological. It doesn't solve the theoretical physics problem. I personally believe in the reality of time and in the fundamental reality of time in physics, but in biology, I think it's a derivative, right? So that's kind of my thinking about it at this point.
I think that a point of view, a possible point of view to attack this problem would be to think to which extent is it convenient for an organism to encode the time locally. So in
Yes.
one unique or elliptical group of molecules that harbor this information in form of concentration or
Yeah.
something like this.
I think that's dangerous because it's vulnerable.
No, the problem is it an advantage or not an
Yeah,
advantage?
I think it's a disadvantage. Yes, because I think it would mean that any mutations in these group of molecules, because all molecules interact with all other molecules, will be detrimental. Of course, it would also be an easy place for evolution to tinker. And we do have some evidence for this. I mean, what I'm convinced of is that it's unrelated to the cell cycle. I think the cell cycle is a derivative of the genetic encoding of time. But for example, a group, proneural proteins that I should, like neurogenin 2 specifically, not talking about this time, but we showed many years ago that all proneural proteins contain the exact same phosphorylation site in exact same position. And if you inhibit the phosphorylation of proneural proteins, they live longer, right? And they will keep cells in a progenitor state longer. And at the same time, they will drive the generation of more neurons, right? So we're starting to get onto nodes, and we're trying now to understand, is there a relationship between APP and proneural proteins, right? So you could be right, and those would be this set of molecules that somehow then feed on mitochondrial metabolism, and that is then the driver. That's a possibility. And the other possibility, which I have to think about whether it's contradictory or not, is what I just told you about the fact that simply the total amount of stuff is time, right? So we'll see.
Encoding these quantities, space or also time, this applies also to positional information. It's exactly
Yeah, exactly.
Encoding these quantities in a limited number of molecules.
You think is advantageous?
Can provide some evolutionary advantage, not on the ontogenetic scale.
Yes, but on an evolutionary scale. Yeah. On the phylogenetic scale.
The advantages of mutations portable.
Yeah. No, that makes sense. There was a question behind you, I think, before.
Pretty simple question, because all the really interesting research you did is for Alzheimer's that's due to genetic
Yes.
mutations, right?
Yeah.
What is actually the percentage between Alzheimer's and
Oh, yeah.
genetic mutations and without?
Oh, yeah. No, but I mean, it's like 99 to 1, right? So
It's always genetic.
no, the other way around. It's almost always sporadic. However, at least in the Western population, there was just a paper out. So basically, there's an allele called, a gene called ApoE. And it comes in 3 flavors, I believe, called 2, 3, and 4. I don't know what happened to 1. And ApoE4, 4 flavors?
Yeah, it's 4.
Is it 4? Is there ApoE1? Say again?
ApoE 1, 2, and 3, and 4.
Okay.
ApoE 4 is homozygous.
No, exactly, yes. So ApoE, the idea is that homozygosity for ApoE4 puts you at high risk, and that risk can be up to 70% of AD. Resequencing, recently this paper just came out, I think in Nature Medicine or somewhere, saying that actually it's 100% of ApoE4 homozygous that attain Alzheimer's disease. So that would be the idea. Now, on the other hand, I know of some emerging evidence from a colleague of mine in Africa that argues that so far, the data they are looking at suggests that ApoE is not at all a locus of risk for Alzheimer's disease.
It's sad to think that you're completely determined by this genetic settlement.
Yes, Well, again, it puts you at high risk. Yeah, I mean, I have to say then, so from our perspective, really, at least my personal perspective, this was never about studying Alzheimer's disease. The idea was a kind of a proof of concept, starting with a simple question: what is this protein doing in humans? And not at any point did it wasn't a joke when I told you I didn't believe my students when they showed me the data. I said, this cannot possibly be. Somebody would have seen this. It's impossible, right? And only after doing, 10 different experiments in 5 different ways that you go, okay, well. So it was really about that, and the whole thing now became about the whole temporal control, and then you start to ask, well, okay, given that this is affected in this disease and so on. Now, what's remarkable, there are 2 ways you can think about the disease. The first is that the terminal pathology that people look at looks similar, right? And so the assumption is that the pathophysiology is the same. But you can fly to Paris via Milano, via Roma, via 10 different places, you still end up in Paris, right? So maybe the pathophysiologies of these different patients is not the same, and maybe this is not one disease, right? That's one possibility. The other possibility is that what the genetic etiology tells us is that all of this is due to some sort of a temporal deficit, and that anything that affects cortical temporal deficit, whether it's environmental or genetic, whether it's APP, ApoE, or some, I forgot now, there's a number of other genetic risk factors that each contributes a little bit, can in the end give you this type of disease, right? So either way, it's about that idea, that concept, the extent to which the vulnerability of the human brain, because it's slow. So if you will, the advantages of slowing down is to give you a cortex with, 16 billion neurons and God knows how many glial cells, and probably the same number of glial cells and so on, and make you able to listen to talks about the cortex and think about them. The disadvantage is that slowing makes you vulnerable to things that, say, a mouse isn't vulnerable to, potentially. And that would be the kind of the bigger lesson, if you will. Yeah. Yes. Yes, absolutely. So basically, this was an idea that we approached. I approached Victor Borel, who works on ferrets, and asked if he would be willing to, and he said yes, and then nothing happened. So we'll see. But I recently discovered that there is a super interesting species of rat, very large rat, that is gyrencephalic, and that has a gestation period of about 150 days and a cortical neurogenesis period of about 70 days or so. So that is a very slow developing rodent, and I would love to look in that, because that's a rodent, right? So now you have and we know that in this species, the markers, the usual antibodies that you use for mouse and human, they also work. SOX2 and CTIP2 and so on and so forth. So I hope within a year or so or more, we have some iPS cells and so on, and we'll try exactly those types of experiments. Yeah.
During the experiment of human progenitor that you showed, did you perform any test on mitochondrial activity or
Yeah, so we're actually looking at that now. We didn't at the time, and didn't really think that why would APP affect it just didn't occur to me, to be honest. And I was thinking very much around those molecular pathways, that we know have an influence of neurogenesis. And when Pierre's papers started coming out, I thought, well, maybe we should look. And now we're looking at this, and what we see is, at least in, so there are 2 links between APP and metabolism. The first, kind of well known, or at least reasonably established, which is cholesterol metabolism. fatty acid metabolism in general, and that, of course, can feed into mitochondrial metabolism. The other thing that we observed going back, so we did EM at the time on these neural progenitors. And the data, I mean, they are just there. It shows you to what extent you don't see something if you're not looking at it, right? I'm looking at these EM pictures when, you know, and I say, okay, well, they kind of look, and we see these vacuoles and so on. We don't know what they mean. And then suddenly you look at the mitochondria, which I didn't 2 years ago, but now I did. And they definitely look different in the knockout versus the control. Very preliminary stuff. Now the question is this cause or consequence, right? So are the mitochondria smaller in the knockout because you accelerated or you increased the neurogenic drive, or is it the cause of, you know, and so that's something we can now look at by manipulating oxidative phosphorylation with, you know, and seeing if we can rescue the phenotype. Of course, that's completely possible, but you're always looking at the control, right? So the process of production of cells should be affecting both equally. And so the difference is presumably due to the mutation you made. And one of the things that actually you see rarely in, I should say, in studies on organoids and so on, and human neurogenesis, which we did in this paper, was to do a rescue experiment. So what we did is take these progenitors, right, they're all knockout for APP, and introduce APP back in a viral vector with GFP. So now we could tell these are the mutant cells, and these are the mutant cells with APP back. And these are the control cells in another dish, right? And basically, the mutant cells differentiated, and the green cells, that is to say the rescued cells, stayed progenitors. So it's in the same medium right next to each other, right? So that told us, one, the phenotype was cell autonomous, and two, it wasn't an artifact. I mean, it was really due to the absence of APP.
In that case, it would be overexpressing, right?
It would be an overexpression, and the funny thing is, they, as I just said, they stayed, I mean, I tried to slide it in, they stayed progenitors, whereas the controls would differentiate a little bit, right? And here, like 0. So in a sense, it pre-configured, you know, the things we're finding in the AD mutant. That was human APP. Of course, that was human APP. So I'm trying to recruit a PhD student now who the idea is, I tried to convince Danya of the experiment. I said, you know what you should do? You should rescue human cortical neurogenesis with fly APP. She's like, no, I don't want to do that. No. So, you know, we're going to do that experiment. Or not rescue, who knows? I mean, then that tells you that, back to the evolutionary question, that the human protein evolved somehow. Well, I mean, yes, that would be great, but it would be very hard. However, because we're measuring, you know, can you just block differentiation with the fly protein or not? If you can't, then, right? At least the protein level. Yes? Exactly. I didn't perform anything. People in the lab did the work. Exosomes? No. Yeah. No, I understand the question. No, we didn't really look at it, although that's another avenue that I mean, it's well known that APP is an endocytic regulator and is itself, as I showed you, by Wnt signaling, its own endocytosis is regulated. And we see in EM these very strange large vacuoles that are larger that are greater in number and in size in the APP mutant. We have no idea what they could. They're probably endosomes of some sort because they're clear, they're empty. They're definitely not lysosomes. And so that could be, at the cell biological level, to get to the heart of your questions, at the cell biological level, What is happening downstream of APP and in its absence, we don't yet know. How is the cell biology of these progenitors changed? We don't yet know. And there's the fat metabolism potential, cholesterol binding. There is the endocytosis, which might be very well linked because the amount of cholesterol on the membrane can regulate how much endocytosis, or can potentially regulate how much endocytosis you have. Could be mitochondria, could be all of those linked to each other. And we don't know the answer to that.
What that you told us was based mainly on neocortical or alveolar precursors. Okay, so what about neural precursors with a different positional identity?
Yeah, so what we did was look at, although I'm not super satisfied with that experiment for a number of reasons, we did look at spinal cord motor progenitors, and we didn't see any difference. So it seems to be rather specific, I mean humans, so the same iPS cells, we just differentiated them into motor neurons, and we saw no difference in the rate of production. And so either that, or it turns out that the protocol for inducing motor neurons from human iPS cells that many people use, and which our colleagues use as well, our collaborators on this, actually includes trying to drive neurogenesis by inhibiting notch signaling. So I don't know if there's no effect or if there's a bit of an effect which then was masked by the way the experiment was done. And that's why I'm not entirely satisfied by the way we did the experiment. Yes? The RNA interactome, as in the binding between APP and RNA? I'm not aware of any evidence that APP is an RNA binding protein. So no, we didn't look. Yeah, and it doesn't contain any domains that would suggest that it might bind RNA. There is some people who argue that the intracellular domain can go to the nucleus and regulate gene expression. I personally don't buy that. We've tried in the past doing a couple of experiments in that direction. I don't believe it. So I don't really think that it's a nucleic acid binding protein in and of itself. On the other hand, who knows? Anything's possible. Any other questions? Okay. Thanks.